Hello, this is Dr. Rinarani Ray. Today I will be discussing on biofilm. So what is biofilm? Biofilm is an assemblage of surface-associated microbial cells that is enclosed in an extracellular polymeric substance matrix. The cells within the biofilm produce the EPS components, which are typically a polymeric conglomeration of extracellular polysaccharides, proteins, lipids, and DNA. Because they have three-dimensional structure and represent a community lifestyle for microorganisms, they have been metaphorically described as cities for microbes. So here we can see how biofilm is forming. The free-floating or planktonic cells of the bacteria, they are trying to get attached on a biotic or biotic surface. After the attachment to the surface, they are trying to form a monolayer with the help of the EPAs, that is extracellular polysaccharides, and they are forming the matrix. And after forming the monolayer, gradually a microcolony is being formed, that is a multicellular matrix. Then the biofilm is gradually growing, and after formation of the mature biofilm, it looks like a mushroom. From this well-grown matured biofilm, some bacterial cells are getting detached and they are trying to find out a new place for their attachment where they can form a new biofilm. So quorum sensing plays a pivotal role in the formation of biofilm. Bacteria produce and release chemical signal molecules called autoinducers that increase in concentration as a function of cell density. It is a regulation of gene expression in response to fluctuation in cell population density. So the bacteria must have three characteristics. First one is to secrete a signaling molecule that is an autoinducer. Secondly, to detect the change in concentration of signaling molecules, that means they can sense the change. And third, to regulate gene transcription as a response. This process is highly dependent on the diffusion mechanism of the signaling molecules. So quorum sensing signaling molecules are usually secreted at a lower level by individual bacteria. At low density, the molecules may just diffuse away. But at high cell density, the local concentration of signaling molecules may exceed its threshold level and trigger changes in gene expression. So let us see. When low density of cells, bacterial cells are remaining present, their individual cells produces acyl homoserine lactose autoinducer, and this autoinducer, it can diffuses the cell wall and it attaches to and activates regulatory protein. This regulatory protein in turn binds to the DNA, that is a bacterial DNA. It triggers the transcription and translation and formation of more and more autoinducer molecule, that is acyl homoserine lactose autoinducer. So more cells means more autoinducer, and this high density of cells, they are producing more and more autoinducers, and as a result, a high amount of cells are there and biofilms are eventually being formed. Biofilms may form on living or non-living surfaces and can be prevalent in natural, industrial, and hospital settings. The microbial cells growing in a biofilm are physiologically distinct from planktonic cells of the same organism, which by contrast are single cells that may float or swim in a liquid medium. Biofilms may form on a wide variety of surfaces, including living tissues, indwelling medical devices, industrial or potable water system piping, or natural aquatic system. Here we can find that biofilm is being formed on an abiotic surface. Here, the gradual formation of biofilm by bacteria are seen. Now, here we can see the pseudomonas cells, the free-floating, the planktonic cells. They are trying to get themselves attached on an abiotic surface to form the biofilm. After attachment, they are trying to form the biofilm, and within 45 hours, the mature biofilms are formed. Here we can see the initial biofilm formation. This line, they're releasing, the encapsulates the sessile bacteria. Some are still free floating, and gradually the structure is increased and it is made up of heterogeneous bacteria. And this biofilm protects bacteria from antimicrobials, and planktonic counts do not correlate with the biofilm. So here it is a complex community of microorganisms. Now here we can see on the biofilm matrix, the glue-like matrix, a number of bacteria remain attached on an abiotic surface. We can find biofilm in various biotic surfaces also, like in the oral cavity, on the cardiac tissue, and also on the implant or the prosthetics. In the oral cavity, the biofilms are formed on the teeth enamel, causing the yellowness of the enamel of the teeth. And if it continues, it will infect the gum and it causes caries and the disease known as a periodontitis. And eventually, the patient is suffering from teeth loss. There are so many diseases that are caused by biofilm. The chronic prostatitis, that is, this formed in the prostate gland. Then otitis media, that means that many children are suffering from pain of ear, that is, biofilm is being formed within the ear cavity. Endocarditis, as the name implies, it is formed on the cardiac tissue. Cystic fibrosis is very well-known disease when the respiratory tissue, the respiratory organ is attacked by biofilm. And as we have discussed, the periodontitis, this is the biofilm formed on teeth. Majority of hospital-borne chronic infections are biofilm associated, like indwelling medical implants. Methicillin-resistant Staphylococcus aureus is formed on these medical implants. They're found on the surfaces in the hospital environment, mainly in the operation theater. They're found in the bathroom surfaces in hospital, clinics, and home, in the air handling and water handling system, in the biological fluid handling machines in dialysis. Also, they cause the chronic wounds in diabetic ulcer, infection like ear infection, and diseases like cystic fibrosis. Biofilms are present on the medical devices. They can be often found on the central venous catheter, intrauterine device, artificial hip prosthetics, artificial voice prosthetics, urinary catheter, and prosthetic heart valve, and so on. Here we can see how the biofilm is being formed on the surface of medical device. It is gradually being covered. Now, the problems caused by biofilms are enormous. Biofilm formation is a critical issue for almost all surfaces in health care and food preparation settings. It can resist antibiotics and affect the immune system of the host. It affects drug permeability. It can form almost any place where water is present, including catheters, surface of kitchen sink, dental enamel, everywhere, almost ubiquitous. Biofilms eventually develop on the interior of most water distribution systems, this biofouling represents a contamination that may present a threat to public health. Here we can see different microorganisms are forming biofilm on the implants and prosthetics, like E. coli, Pseudomonas aeruginosa, Staphylococcus aureus, Staphylococcus epidermidis, Serratia, Proteus, and fungus like Candida used to form biofilm on contact lens. On the other hand, we can find Pseudomonas aeruginosa, Enterobacter species, and Klebsiella species on the catheters. In the hard vein Streptococcus species, Staphylococcus aureus, Staphylococcus epidermidis, Enterococcus, and Candida are found. And on the hip joints, prosthetics, Staphylococcus aureus, E. coli are very common microorganisms. So clinical and public health microbiologists recognition that microbial biofilms are ubiquitous in nature and has resulted in the study of a number of infectious disease processes from a biofilm perspective. Among all microbial and chronic infections, 65% and 80% respectively are associated with biofilm formation. Bacterial biofilms show resistance against human immune system as well as against antibiotics. So adoption of new strategies to combat biofilm formation and biofilm forming organisms and their application to cure human health and disease become the need of the hour. So be very careful from biofilm. Thank you.